joining us for this first virtual Clay Minerals Society workshop. Today I'm going to present our research on antibacterial clays, and this is the first results that we've had testing the clays in vivo in mice infected with MRSA. The animal testing was all done at the Infectious Disease Research Lab at Mayo Clinic in Minnesota with co-author Susie Schmidt-Malan conducting the lab work and Robin Patel, who is supervising. So I'm going to start by reviewing some of the past results of testing the antibacterial clays in vitro so that you'll have the background for understanding what's happening in vivo. Unfortunately, we were not able to get any antibacterial action in the wounds. So I'm going to present hypotheses for why this may be happening so that maybe we can develop better methods for applying clays to infected wounds. So this work began in 2002, approximately, when members of the Clay Mineral Society were contacted by this woman, Lene Brunet de Corso, who is treating these children in the Ivory Coast of Africa who suffered Buruli ulcer, which is a flesh-eating disease. She treated them with clays that she got from France, where she grew up, using clays on bites and wounds. And this is a graphic image of how severe this infection is, but by applying clays daily for over 3 months, she was able to heal these infections. And at that time, there was no antibiotic that worked on these infections. So this is what got our interest in that clays are actually doing something, and we need to figure out what it is. We couldn't find the geologic source of these clays in France, but we were able to find clays in the US, in Oregon, that were similarly antibacterial. This is the Oregon blue clay. I call it the Oregon Mineral Technologies clay because that's who owned the deposit and allowed us to do the work on this mine. Here's the blue clay shown here, and it is an altered porphyritic andesite. This is shown in the top right here, and in B and C, those are actually completely turned to clay. It's a hydrothermal alteration of this andesite. So where are these formed? They're formed in geothermal systems around the Circum-Pacific, wherever there are there's volcanism and volcanic massive sulfide deposits are forming. So this is the argillic zone around a porphyry deposit. There was unfortunately no copper or gold in this deposit, but there's lots of iron and there's lots of sulfur. Through isotopic studies, we determined that the clays were formed at between 100 and 300 degrees C. They're very acidic. From 2 to 5 is the pH of a clay suspension. The clays are primarily illite, smectite, and pyrite. So here is the main pit of the mine where this Oregon blue clay comes from. And this is Keith Morrison down in the hole digging away. He is a PhD student that worked for me on this deposit. It was his dissertation. So most of this work that I'll be presenting is his work. And he did a fabulous job of mapping all of the clay types across this deposit and correlating them with their antibacterial activity. So this red clay on the surface is oxidized blue clay, but the blue clay is just about a foot underneath the surface. There's a lot of faulting in the area, and we believe, and there's white clay that is smectitic along the fault zones. We believe that those were in place by meteoric fluids. The clay alteration was 30 million years. So there was plenty of time for these clays to form. At the bottom, there's stockwork that is full of quartz and pyrite. In 2008, we worked with Dennis Eberl on this deposit initially, and this is the X-ray diffraction of the main clay, the blue clay, and this is the less than 1 micron fraction oriented clay mounts with copper K alpha radiation. What Denny recognized is that what was previously identified as pyrophyllite is actually rectorite, as shown here. And the other interesting thing about this pattern is that there's considerable pyrite in the less than 1 micron size fraction, which is probably important to its antibacterial effect. So I know that there are some people who are not clay mineralogists who are listening. So I wanted to just show what rectorite is. It's a 1-to-1 ordered illite smectite. And what we mean by that is there's a tetrahedral, octahedral, and tetrahedral layer. That's the 2-to-1 layer. The smectite has a space between the layers, which is expandable. So you can get hydrated cations going in that space. Illite, on the other hand, has that space it collapsed, usually around potassium, and so the charges are satisfied in the illite. So this clay is 50% smectite, 50% illite. One thing that's important is where is the iron? And the iron can be in the octahedral sites of these clays. It can also be in the interlayer sites of the clays, and it's important to differentiate those two. Wanted to introduce the morphology of the iron sulfide in this deposit. In the stockwork, most of the clay contains pyrite that is greater than 25 microns. In fact, this is a chalcopyrite, and those are not antibacterial. There's no antibacterial activity when we separate those large pyrites, but there's spherical pyrites that are embedded in the clay matrix shown here. This is a 3 micron scale. And if you look closer, there are lots of these little spheres that are iron sulfide rich. And this is zoom in to 250 nanometers, showing that it looks pretty much like a bacterium that is probably precipitating out iron sulfides. We think that these are the more reactive pyrite in this system, being the supplier of the iron 2 that's high in this area. So if you take that clay and you suspend it in water, and then you apply it to bacteria in their log phase, this is what they look like over time, from initial to 24 hours. And you can see it after 1 hour, there's a beginning of black precipitates at the ends of the bacterium. This is E. coli. It's more pronounced after 6 hours. You can see a clear polarization of these black precipitates. And then after 24 hours, they've gone inside the cell, or they appear inside the cell. The white areas in this photograph are phosphates that have been plucked out during sample prep. So we were wondering, what are those black particles? And Keith Morrison in 2014 did electron energy loss to determine that those black particles, which appear white in these images, were actually iron 3 oxides. So the mechanism that he came up with was that the iron in solution, which is quite high, is reacting with oxygen when we add the oxygenated water, and through Fenton reaction series is producing hydroxyl radical. Sorry, I've lost my pointer. There it is. Hydroxyl radicals shown here. But hydroxyl radicals only exist for nanoseconds. They're an intermediate product, and they react very quickly with anything within nanometers of its site. So we think that the iron is penetrating the cell wall, going in as iron 2 to the intracellular sites, then reacting with proteins and DNA in the cells and precipitating these iron oxides. Keith and my other student, PhD student, Carolina Londoño, used secondary ion mass spectrometry imaging of the cells to image where the aluminum and iron went. And this is an image of the E. coli cells. The iron is red and it's inside the cells, and the aluminum is yellow and is clearly outside of the cells. Carolina selected one E. coli cell and did a cross-section, and this shows the aluminum relative to CN ratio. In the red is the treated cells, and the blue is untreated cells. And you can see that the aluminum is high on the cell walls, whereas the iron is high in the interior compared to the untreated E. coli. So the bottom line of Keith's work was that he had sampled all over this mine and found out which clays were antibacterial and which were not. He found that the clays that are antibacterial, shown in this blue shaded area, are in the iron iron 2 solubility field. But once you get to high pH, goethite precipitates, and those clays are no longer antibacterial. Similarly, for aluminum, the antibacterial clays are in the aluminum field, and as soon as they cross out of that field at about pH above 5, they precipitate out as iron 3. Okay, and later on, we're going to use the red clays in this area that are mineralogically similar to the blue clay, but not bactericidal because of their oxidation state. So you might wonder why we don't just put water in the wounds and treat the wounds with these iron and aluminum in solution. So Keith compared the aqueous leachate of the clays to the clay suspension over time. This is over 24 hours, and showed that iron 2 decreases rapidly without the clays present. But when the clays are present, I believe it's because they're buffering the pH of the solution to conditions where the iron 2 stays stable. So the iron 2 is high, and aluminum is high as well compared to the leachate. He also studied the peroxide as an indicator of hydroxyl radical formation, and you can see that the leachate loses its hydroxyl radicals immediately, whereas when the clay is present, it stays fairly stable for 24 hours. So we went to Mayo Clinic and teamed up with this infectious disease research lab. They first wanted to test all of the bacteria that we had tested and their own bacterial pathogens against biofilms, because biofilms are much more common in the wound, and they are better at protecting themselves from destruction. So they did a biofilm study the first year and basically grew their bacteria on polycarbonate membranes. Then we applied the clay in suspension, and we varied the concentration of the clay between 100 and 800 milligrams. Testing how well it worked at a high concentration versus low concentration that we might need to use in the animal trial. And then standard plate counting was used to determine the bacterial population. The blue clay always killed the bacteria. These are the results of their survey, and you can see there are 12 different bacteria in the collection that were studied. Gram-negative and gram-positive bacteria. Here, the red bars are the bacterial growth after 24 hours. The blue bars show the application of the blue clay, and the green bars represent the aqueous leachates. In every case, the blue clay and its leachate reduce the bacterial population significantly, and even more so for the gram-positive bacteria. So here is a Staph aureus IDRL 6169. That is a methicillin-resistant Staph aureus that we chose for our animal trials. This shows the time kill curve for these for the MRSA. The bacteria grow over 24 hours, shown in black here. The leachate had very little effect on reducing the population in this study, although it was bacteriostatic. But importantly, the blue clay reduced the population in less than 10 hours at 50 milligrams per milliliter. So this is the mouse testing. We used Wistar mice. They were sedated with ketamine and xylazine and had some pain management. The fur was shaved and disinfected. The skin was punched with a biopsy punch of 5 millimeters. It was then infected with the MRSA at about 10 to the 8th colony forming units per mil. The wound was treated with the hydrated clays and controls for 5 days with the dressings changed daily. The dressings were covered with hydrogel and gauze, and I'm going to talk about that more soon, and fastened with Tegaderm. Then the infection was evaluated by standard tissue cultures in colony forming units per square centimeter tissue. And these are the results. We tested different doses of the clay. We used a large amount of clay, the 800 milligrams, to stabilize the clay in the wound, because the mice move around, and when we were working with 100 or 200 milliliters, it was not possible to keep in the wound site. But 800 milligrams may have been too much, reducing the amount of water available. We went down to 200 milligrams, and then we settled on 400 milligram doses. But you can see in every case here, there is no significant change in the infection between the untreated, Vaseline treated, the OMT Oregon red clay, and the Oregon blue clay. All of them failed, except for bacitracin was successful in a few trials at killing the bacteria, but otherwise it didn't work. Other parameters we looked at were the wound diameter, and you can see there's basically no change in the wound diameter during the 5 days, the healing process. Each of these dots is a mouse, by the way. There's three tested for each. Purulence was tested, and we did see an effect of the OMT red and OMT blue clay on the purulence. It was reduced compared to the other controls. However, we think that's just because there's smectite in those clays, and the wound exudates are absorbed by the smectite. So what's happening? Why didn't it work? We had 4 hypotheses that we went to test. First was that the pH and EH of the aqueous suspension of the clay was out of the stability range for iron and aluminum. That's limiting the antibacterial reactions. Second is that the iron and aluminum, or their products, Fenton reactions, might be complexing with components of the wound. The clay could be drying. In fact, we did see that it was drying in the beginning, and so that's why we worked on developing a way of keeping it moist during the treatments. And then finally, that the supply of oxygen to the wounds might be diminished relative to the in vitro experiments that were successful. So we first tested the hypothesis that the pH and EH were changing. I tested the OMT blue clay over 100 hours for pH and EH, and compared that to the Clay Mineral Society Illite IMT1 and Smectite SWY1 pH and EH over time. And you can see that the pH didn't really change significantly over 100 hours in any of the clay samples. But the EH does change in the first 10 hours in every suspension of illite smectite, illite, and smectite. There's an increase over 10 hours, and then it gradually decreases over time. But, oops, sorry, Beck. So we also tested the change in the pH and EH after reaction with the MRSA, and this shows that the pH doesn't change, but the EH is definitely decreasing after treatment with just the bacteria in solution. So planktonically, we tested also wound fluids, simulated wound fluids, which here are made of fetal calf serum, sodium chloride in 0.1% peptone, and also sheep blood. And these results show, the black bars show the growth of the MRSA over 24 hours. The red bars show the experiments with the red clay, and you can see it does not affect the bacterial growth, but in the presence of the blue clay, bacterial growth is definitely defeated. So it's working in the presence of those planktonically. So what about biofilms? Well, the biofilms shown here, the red clay is in red and the blue clay is in blue. The red, here we mixed the clays and MRSA with a peptone and sodium chloride and the fetal calf serum and then the blood. And for the sodium chloride and peptone, there was no significant effect on the results, but the antibacterial action was impeded in the presence of fetal calf serum and with blood. So it is possible that there's something complexing in the wound environment. This was our experiment on hydration state. We made a hydrogel with sterile xanthan gum in deionized water and added that on top of the clays. Here I'm just showing that there's no difference in the antibacterial effect of the blue clay suspended in hydrogel versus deionized water. There is a slight increase in the EH in the hydrogel, which can only help in this situation. And finally, we tested for the oxidation state change in the presence of various components, one at a time and in combinations and with MRSA. And so to sort of summarize all of that data, this diagram shows the EH of the blue clay in water, but in the presence of the simulated wound fluids and blood and bacteria, the EH is dropping by about half. The pH is not affected at all. The clay continues to buffer the pH to between 4 and 5. And the red clay also, the pH is 7 to 8, 8.5 range, but there is a reduction in the oxidation state when reacted with the MRSA. So in conclusion, we showed that the colony biofilms, including drug-resistant bacteria, are killed with the antibacterial clay in vitro, but the antibacterial clay is not effective on the mouse wounds treated daily over a week. Reduction of the mineral reactants in the wounds may explain the diminished effectiveness in vivo, and complexation of reactants is possible. But we recommend enhanced oxidation of the clay in vivo to improve the wound healing. Thank you. I'll take questions.
Thank you very much. Are there any questions, please? Anybody wants to ask a question? Yes, please.
Thanks, Professor Williams. I've got a very small question. I'm quite curious about how you guys start up the animal test, because you said that the clay suspension, if it is very diluted, it's not possible to put the clay suspension with the mouse because they are moving always. I mean, you use the concentrated clay suspension and then put them onto the bandage and then bound it to the mice, or how do you do that? I mean, when they're moving,
yeah. First of all, they're sedated when this is happening, so they're not moving when we're applying the clay. But the wound is infected, and they make sure that the infection is established, and then put the suspension. And we came down to using 400 milligrams per milliliter. So it's still, you can pipette it into that wound. And then it's covered with the hydrogel to keep it moist. And then we cover it with gauze, just regular gauze, and is attached to the skin with Tegaderm. And Tegaderm is an adhesive that allows oxygen to diffuse through that dressing. First, we were using just the Tegaderm, and then I found papers suggesting that Tegaderm isn't all that great at diffusion of oxygen. So we thought that oxygen was being limited. But even when we just used the gauze and attached it with the adhesive, we're still seeing reduction in the wound.
Okay, thanks. And very tiny extra question is about you said that it is the reduction of reactants in the mouse test that causes ineffectiveness. But in the in vitro test, actually, if the clay wants to be antibacterial, the reactants definitely be reduced due to the thermal reactions. Right.
Yeah, in vitro, there's a lot more oxygen, is all I can say. There's a lot more oxygen in the fluids. But when we put it in the wounds, it's either the serum of the wound or perhaps the hemoglobin that may be complexing with the reactants, or just the oxidation state is not high enough. And so the first thing to try is adding an oxidant to that application to see if the clay will perform as it should when it's in the right oxidation state.
If it is possible to quantify the oxygen production in the mice test, I mean, from the serum suspension stuff.
Certainly, I think that you could. Well, we did measure the pH and EH before and after in the clays that were taken off. There could be ways of measuring the oxidation state in the wound itself, which we have not tried.
Okay, thanks.
Okay, thank you. Any other questions, please?
I have a quick question. Linda, you have a nice talk. Do you think which component in the blue clay plays a leading role in changes the reduction of mineral reactions with bacteria, and for the
I'm sorry, missed that. Which, you're asking what company?
Yeah, which components in the mineral in the blue clays plays a leading role?
Oh, which components?
Yeah.
Okay.
Smectite or rectorite also?
No, it's the rectorite. Well, the clay contains pyrite and rectorite and a little feldspar and quartz. We think that it's the clay because our comparison of just the leachate to suspensions with the clay present, we think that the clay may be just there buffering the fluid composition to a pH EH range it needs to be, or at least the pH range, and it also may serve, if the reduced iron is in the interlayers of the clay, it could be a reservoir for that reduced iron to gradually release that, like a time release capsule. But there's also clay iron in the octahedral sites, and there has to be dissolution going on because we have high aluminum and high iron too in solution. So there's some dissolution happening as well.
So the chemical composition also is important. You mean?
Chemical composition. Well, absolutely. You have to have the reduced metals. You have to. My second PhD student, Carolina, showed that the aluminum is key in the destruction of the cell membranes as well. It's binding to phospholipids in the membrane, causing them to open so that the reduced metals can go into the cell. So all of those components have to be there. But it doesn't have to be a rectorite. It can be any other clay that contains reduced iron.
Okay, thank you.
Okay.
Thank you very much, Linda.
Raoul, it's Kevin here. There's a question in the chat, actually.
I see. There is. Sorry about this. So there is another question. Please tell again, if I missed it, the name of the organism in the disease treated in the field. So this is the question from Robert Jarrett.
Oh, in the French green clay, that it was Beruli ulcer. It's a Mycobacterium ulcerans, very slow growing bacteria, but it eats the fatty tissue under the skin, and then the skin falls off and exposes the muscle and bone underneath.